All right, this is the third time recording this, so hopefully it works. My name is Jacob Bradshaw, and my presentation is on the evolution of the endosymbiotic theory. Now, before we go through it, to give you some preface and some outline, we're first going to talk about what the theory is and really basic what it is. Can I show you a diagram of how that process might have occurred? And then we're going to go through the history and the scientists that made the breakthroughs to, prove that theory and to help it to evolve into what it is today. So first, to start out, what is the symbiotic theory? Now, the endosymbiotic theory is also known as symbiogenesis, and it simply is an explanation for the origin of eukaryotic cells other than random mutation. And it focuses on several key organelles of eukaryotes and how they originated as a symbiosis between separate single-celled organisms. Some of those organelles are mitochondria and chloroplasts. Now, this would have happened about 1.5 billion years ago. This symbiosis would have taken place. And this would have taken place or arose from proteobacteria and or cyanobacteria. Now, proteobacteria would or could have become the mitochondria, and cyanobacteria the chloroplasts. Now, the process that would have happened, as you can see in the picture where with three, we have this larger cell that's growing that begins a symbiotic relationship with this purple bacteria in stage 3. And then in stage 4, you have this phagocytosis that occurs to keep that symbiotic relationship going, and if the bacteria move. And now it's, considered an endosymbiotic relationship. Now, once this phagocytosis occurs, we're going to run into what's called gene transfer, where we have pieces of the bacterial genome being incorporated into the host cell's genome and DNA. Now, most of their genes are going to be transferred to the host cell. Now, to give you some numbers, cyanobacteria and proteobacteria today maintain a large genome, greater than 6 million base pairs, whereas plastids, mitochondria, and chloroplasts have a very reduced genome size. Chloroplasts in photosynthetic organisms are normally between 120 to 200 kilobase pairs that code for about, between 220 and 200 proteins, whereas mitochondrial genomes in humans are approximately 16 kilobase pairs that code for 37 genes, and 13 of which are proteins. Now, it's proposed that this gene transfer, or the loss of most of the plastid's genome, actually happened very rapidly or very early on in the development of this endosymbiotic relationship. Now, over time, that host cell would engulf more as it need be. But that's really the theory. It's saying, hey, eukaryotic cell developed from simple prokaryotic cells that were engulfed into a larger prokaryotic cell and ultimately cooperated to form what we are today. Now, to get into some of the scientists that contributed, the first of which is Andreas Skimper around 1883. Now, he coined the name of chloroplasts. He was studying plant cells and seeing these. Now, what's pretty cool, though, is that he saw that chloroplasts, when the cells divided, would actually divide on their own in a process similar to binary fission that bacteria and cyanobacteria do. So he saw this and saw that they resembled bacteria, and kind of proposed, or I guess planted the seed of the idea that, you know what, these may have not originated inside the cell, that they might have actually come from outside the cell. Maybe they were a bacteria. Now, stepping on his shoulders to take it further was Konstantin Mayakovsky around the 1920s. And he really actually proposed the idea that plastids didn't come from outside of the cell, but they were actually endosymbiotes. And this idea kind of made a rivalry between symbiogenesis and Darwinism, saying that the origin of the species didn't actually occur from random mutations, but symbiosis. Now, Mayakovsky went on with Ivan Wallin and studied bacteria and wrote books and proposing this idea. They ran into a problem, though, because they didn't have the technology to prove that these plastids originated outside the cell. And so they kind of run into a problem there. Others were studying around the same time, and one of which was Boris Polanski. And his quote is, "The theory of symbiogenesis is a theory of selection relying on the phenomenon of symbiosis." And he really studied and trying to piece together the idea of symbiogenesis and Darwinism. Now, the next biggest great breakthrough in the theory was Stocking and Gifford. Now, the wall that Mayakovsky and Wallin ran into, where they couldn't prove that these cells originated from outside the cell, that they were not built already in the cell, was that they have DNA. And that's what Stocking and Gifford saw. They saw a radioactive thymidine inside the cell with the technology that they had during that time. It proved that mitochondria and chloroplasts had their own DNA, that they were not actually directed by the eukaryotic genome. They could function by themselves. Ultimately, of course, now in our daytime, they would not be able to function without each other. But it proves that they could have at one point in the past. And so that was a huge step forward. Now, the next scientist was the one that brought a huge step forward and really brought the idea out of the dark and found, I guess, the most proof for it. It was definitely helpful that we have the technology that we did. And this was around 1980s. And this was Lynn Margulis. Now, she was studying, of course, the same thing, but she hypothesized that eukaryotic cells came from communities that just interacted together. And through her studies, she saw that free-living bacteria would actually merge with larger life forms to create something new. She argued against that common idea of random mutation, and that the DNA in eukaryotic cells originated or came from bacteria and not mutations of genetic drift. Like I said, she saw that microorganisms have a tendency to merge. Symbiogenesis is the best explanation for the similarities that scientists see between eukaryotic cells and prokaryotic cells. Like we've studied in this class, and I guess the 5 kingdom, I guess, grouping method, and all the different ways that people categorize organisms here on Earth, we can see that eukaryotes and bacteria, or prokaryotes, have a lot of similarities. Now, Margulis went on to actually argue that eukaryotic flagella potentially came from spirochetes that of course, merged with the larger life forms. And this is harder to prove because in flagella you're not going to see DNA, but all of that prokaryotics DNA would have been incorporated into the host cell's genome. Now, this is really cool. There's tons of other breakthroughs that they made, seeing that the mitochondria and plastids are, I guess, form on their own process, similar to binary fission. They're also seeing that the cell's mitochondria or chloroplasts, if a cell's mitochondria or chloroplasts are removed, then the cell does not have the means to create new ones. Some mitochondria and plastids contain single circular DNA molecules that are similar to the DNA of bacteria as well. And mitochondrial and plastid ribosomes are very similar to bacteria as well. So there's tons of evidence that shows that these plastids actually came from outside of the cell, which is really cool. Now, in conclusion, we can see, and it's not a complete, I guess, rival to Darwinism. They're pieces of the same puzzle, and we just can't see the whole picture yet. We can see that the idea of symbiogenesis can explain how more complex cells were created and originated from, that allowed larger life forms, animals, to form. And then it kind of goes on the same thing. It's like, we know that evolution doesn't occur in a single individual, but in a population, in a species, as different elements are, I guess, forced upon them. And so they are there to create a bigger puzzle. There's pieces still out there that we don't know how everything fits, but I hope that we find those, and it's really there to create the bigger picture of this creation that we see all around us.